Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on chemiosmotic hypothesis. Chemiosmotic hypothesis was proposed by a scientist, Peter Mitchell. So, chemiosmotic hypothesis explains the synthesis of ATP in photosynthesis through a proton gradient that established across the membrane. Okay, so it explains how the ATP are generated by using proton gradient. Okay, so when electrons they are passing through the electron transport chain. Yesterday we have already discussed about cyclic and non-cyclic photophosphorylation. So there the electrons are passing through the electron carriers. So they releases energy. That energy is used up by the ATP synthase for the production of ATP. Okay, let's see what is actually happened in the non-cyclic electron transport. So when light falls on PS2, it is get excited. It releases two electrons. They are transported to plastoquinone and then to cytochrome complex and then to plastocyanin. Finally, they reached to PS1. So, PS1 also further excited by taking the sunlight. It also releases electrons. They are passed to ferredoxin and then to they reaches NADP plus reductase to synthesize NADPH. Okay, so it is actually happened in the non-cyclic electron transport chain. While the electrons are passing through these complexes, energy is released. That energy is used up by the ATP synthase that facilitates the synthesis of ATP. Let's see how the energy is released. So energy is released in the form of protons. Okay, so let's see the proton pumping. Let's see the proton pumping. Let's see how the protons are generated in the electron transport, how they accumulated in the lumen, how they come back to the stroma. Let's see in detail, okay? Let's see how the proton gradient that facilitates the ATP synthesis. So when PS2 get excited, it releases two electrons. They pump to cytochrome complex and then to PS1. When the electrons are passing through the cytochrome complex, it pumps protons from the stroma to the lumen. Meanwhile, photolysis of water occurs. So it releases two protons into the lumen because oxygen evolving complex is located in the lumen. So it also gives two protons. From stroma to lumen, protons are pumped through the cytochrome complex as well as photolysis also gives two protons into the lumen. So now high proton gradient is accumulated in the lumen side, whereas low proton gradient is established in the stroma side. So it creates a proton concentration difference. So that difference, so when the difference is established between stroma and lumen, when the difference is established between the stroma and the lumen, the protons tend to move into the stroma side. So the movement of protons is facilitated by an enzyme complex that is located on thylakoid membrane. So that enzyme complex is nothing but ATP synthase. So this is the ATP synthase. It consists CF0 particle and CF1 particle. This is the channel that facilitates the movement of protons into the stroma again. It facilitates the proton movement through the lumen to stroma. Okay. So when the protons are passing through this ATP synthase, it facilitates the facilitated diffusion occurs through this enzymatic complex. When the protons are moving from higher concentration to the lower concentration gradient, they are coming with energy. That energy will be utilized by the ATP synthase molecule, ATP synthase complex that facilitates the synthesis of ATP from ADP plus inorganic phosphate. One inorganic phosphate combines with ADP results in the formation of ATP. So energy is provided by proton gradient. Okay, so let's see the second important thing, that is PS1 releases electrons. They are moved to ferredoxin and then to the NADP reductase enzyme complex. So it is located near the stroma side. Okay, so then it receives electrons from PS1 as well as it receives two protons from photolysis of water. So it gets reduced into NADPH. Okay, so in this way ATP preparation and also NADPH preparation occurs in the non-cyclic electron transport chain. So these two are called as assimilatory power. So this is the energy used up by the CO2 molecule for the conversion of carbohydrates. Okay. So this is about chemiosmotic hypothesis.